Decoded neurofeedback. Blindsight may be a very useful, or maybe even unique way of dealing with the signal strength confound. For stimuli presented to the blindfield, subjective experience is lacking, and yet patients seem to have a perceptual signal that is pretty strong. One can attend to this unconscious signal pretty much as well as one can attend to a conscious stimulus. But blindsight patients are rare. It involves a lot of meticulous work to establish that they are truly unconscious in the blindfield, rather than just saying that they don't see so clearly after a brain lesion. Researchers often disagree whether a certain patient really is blindsighted, or that vision is just weak or impaired after some cortical damage. So what should we do? Ideally, we need a method to induce strong, unconscious perceptual signals in subjects without brain damage, so that we can easily study them as a group. Recently, a new method based on fMRI was invented, which may achieve just that. This is called decoded neurofeedback. Usually, we think of fMRI as a way to measure or read out brain signals. But actually, with a twist, we can use it to manipulate brain representations too. This is based on a general phenomenon called biofeedback. The idea is that if we measure a certain physiological signal of your body, for example, your heart rate, and we present that measurement visually to you in real time, say on the computer screen, over time, by looking at that signal, you can learn to control it to some extent. The evidence for this kind of phenomenon is sometimes a bit mixed. It seems to work, but again, it is an issue of how robust it is. When applied to the case of fMRI, many studies reported that it works, but there is one concern. Remember that fMRI measures the blood oxygenation signal, which doesn't really change that much. The range of change is on the order of just around 1%. We can see this kind of change when we average the signal over many trials. But when it comes to controlling it in real time, we can't average the measurements. So there may be some limitations on how much we can monitor and control with this small signal. One way to get around this problem is to capitalize on the spatial resolution of fMRI to identify fine-grained spatial patterns of activity within a region. For example, if we present some red lines and green lines to subjects, they may activate the visual areas at roughly similar levels. This is true in area V4, even though we know that individual neurons in the areas may be tuned for different colors. On average, the area will show similar bold signal when red lines or green lines are presented. But if we look at each individual voxel in V4, some voxels may show a small bias in favor of red lines, and some may show the opposite bias. So overall, there may be a spatial pattern within V4 that we can recognize to predict whether the subject is seeing red lines or green lines on a particular trial. This kind of analysis is sometimes called multivoxel pattern analysis, or MVPA. Or sometimes we say we are decoding these voxel patterns using methods from machine learning or artificial intelligence. These decoded signals can be pretty robust. Say we are using them to predict if someone is looking at green lines or red lines. With current methods, we can get it correct easily up to about 80% of the time. So Kazuhisa Shibata did a groundbreaking study combining MVPA and neurofeedback. He did this together with a team of very distinguished Japanese cognitive neuroscientists, Mitsuo Kawato, Yuka Sasaki, and Takeo Watanabe. In the study, they monitored for the decoded multivoxel patterns in real time. If the pattern showed that the brain was likely representing a certain stimulus, they gave a visual feedback to the subject. A circle got bigger, meaning that the multivoxel pattern in the brain looked like what the experimenter wanted, and the subjects would get a monetary reward on the order of a few cents. Over many trials done over a few hours, subjects can earn up to tens of US dollars. That is, they learn to activate the specific voxel pattern that the experimenters wanted. This happened even though the subjects were never told what the voxel pattern represented. They were just told to try to figure out what to do in their minds somehow to earn as much money as possible. Now, here's the most remarkable thing. You may think, given that they learned to activate the voxel patterns, the subjects must have figured out what this pattern represented. But actually, no. They had no idea. They just thought they were playing this mental slot machine. Sometimes they thought they knew, but they were mistaken. When asked, they would say, "Oh, yes, every time I thought about lunch, the circle got bigger, so I would get the reward," or something like that. In fact, the experimenters went further to double-check whether the subjects may know something. So after the experiment, the subjects were told about the real purpose of the experiment. It was explained that the target voxel pattern that would earn them the most reward either represented one visual stimulus or another. They were asked to guess which one it was. But as a group, they were at chance in answering this question. They really had no idea. If we think about it, perhaps it all makes sense. The brain is known to show highly structured, spontaneous activity. So for the neurons representing red lines, every few seconds they will fire more than the neurons representing green lines and vice versa. Our neural activity is never stable. But of course, we are not consciously experiencing the fluctuations of these red and green representations in everyday life. That is to say, there must be spontaneous neural activity in the brain that is meaningful and yet unconscious. Shibata's study probably tapped into exactly this kind of unconscious brain activity. If this seems a bit mind-boggling, it is. We will go through some concrete examples of how we can use this method, called decoded neurofeedback, to test how unconscious signals in the brain may facilitate powerful cognitive functions.